The following are the contents of the topic: the advantages of cross-pollination. Cross-pollination increases the variation in offspring. The following are the 4 main advantages of cross-pollination. Firstly, the new plants that produced from cross-pollination are healthier and more resistant to diseases and pests. The second, new varieties of plants may be produced. The third, seeds produced are with the improved quality and more viable. The fourth, better yield and quality of fruits are produced. Now, we discuss the innovation of cross-pollination. In the field of agriculture, in order to improve the quality of agricultural product, 2 parent plants with good qualities are cross-pollinated to produce offspring that inherit the good, desirable qualities. In Malaysia, innovation of cross-pollination is implemented in the following agriculture activity. In oil palm, the cross-pollination between Dura sp. and Piscifera sp. has produced Tinara sp. which gives higher yield of oil. For the paddy plant, 2 types of paddy are produced using innovation of cross-pollination. MR103 is produced which have higher quality rice grains. MR211 is produced which have shorter maturity period. The third is maize plant. Masmadu has a bigger cob and sweeter corn. The second stages of the plant reproduction is fertilization process. We will discuss fertilization process in plant in this topic. During pollination, the male and female gametes are brought together by their pollination agents. After the pollination, fertilization will occur, at which the male gamete carried by the pollen fuses with the female gamete in the ovule. The fertilization process in plant after the pollination process will go through 3 stages. The first stage is pollen tube generation. When the pollen grain landed on stigma, the sugary fluid on the stigma stimulates the pollen grains to germinate. Each pollen grain produces a tube called pollen tube. The second stage is pollen tube development. The pollen tube carrying 2 male gametes grows down through the style and get into an ovule in ovary. An ovules contains a female gamete. The ovules are found in the ovary of flower, and inside each ovule contains the female gamete. The third stage is fertilization. The pollen tube enters the ovule through a tiny pore, micropyle. The end of the pollen tube bursts to release the 2 male gametes into the ovule. Fertilization occurs when the nucleus of one gamete fuses with nucleus of the female gamete to form zygote. After fertilization, fruit and seed will be developed. 3 stages of development of fruit and seed. The first stage is drop-off of the flower. The petals, sepals, stamens, stigmas, and style of the flower will wither and then drop off. The second stage is formation of fruit. The third stage is formation of seed. The zygote will divide and develop into an embryo. The ovule will develop into the seed inside the fruit. If the flower with more than 1 ovule, more seed will be produced. These are the 3 stages of development of fruit and seed. Now the structures of seed and their function to be discussed. There are 2 types of seed. The first type of seed is monocotyledonous. This is the seed with single cotyledon. The second type of seed is dicotyledonous. This 2 types of seed will be detailed in the following part of the video. The first type of seed, monocotyledonous seed, the seed with single cotyledon. A mature monocotyledonous seed contains an embryo, which consists of cotyledon, plumule, and radicle, which surrounded by endosperm and enclosed by seed coat. The following are the structure of monocotyledonous seed and their functions. The seed coat, it function to protects the seed. The plumule, it will grow and develops into the shoot system. The radicle, it will grow and develops into root system. the cotyledon, and endosperm, stores food which is used by embryo during the germination. This is about the discussion of the structure of monocotyledonous seed, and their functions. About the dicotyledonous seed, the seed with two cotyledon. A mature dicotyledonous seed contains an embryo, which consists of cotyledon, plumule, and radicle, and enclosed by seed testa. Dicotyledonous seed consists of hilum and micropyle that can be seen from the outside of the seed. The follows are the structure of dicotyledonous seed, and their functions. Testa, the hard external coating of a seed. It functions to protect the seed. Micropyle, the small pore which is present just beneath the hilum. It functions to allow air and water to enter the seed. Hilum, a scar or mark left on a seed coat. It functions to attaches the seed to the wall of ovary wall. Plumule, the part of a seed embryo that will grow and develops into the shoot system, bearing the first true leaves of a plant. Radicle, the part of a seed embryo that will grow and develops into the root system of the plant. About cotyledon, the dicotyledonous seed consists of two cotyledons. It functions to stores food which used by the embryo during the germination. This is the content about the structure of dicotyledonous seed, and their function. We continue the lesson with topic, germination of seeds. Germination is the growth process of a seed into a seedling. There are 6 stages to complete the germination of seeds. At the first stage of seeds germination, the seed absorbs water and air through the micropyle. The second stage of the seed germination is softening of testa by the water. The third stage of the seed germination is the growth of the radicle, where the testa bursts and the radicle grows downwards to form the root. The growing embryo uses the food that's stored in the cotyledons, or endosperm. The fourth stage of the seed germination is the growth of the plumule. At the fourth stage, the plumule grows upwards to form the shoot. The growing of embryo consume the food stored in cotyledons, or endosperm. The fifth stage of the seed germination, the cotyledons decrease in size, because the food stored in cotyledons, or endosperm are used up. The sixth stage of seed germination is the process of the seedling begins to carry out photosynthesis, where the plant is capable to make its own food via the photosynthesis process. This is the 6 stages of seed germination. There are 2 types of seed germination. The first is epigeal germination. During the epigeal germination, the cotyledons are carried above the ground. Example of plant seed which germinated via epigeal germination are mung bean, gourd, and sunflower. The second type of germination is hypogeal germination. During the hypogeal germination, the cotyledons remain under the ground. Example of plant seed which germinated via hypogeal germination are corn, peas, and wheat. The following content is the topic about the conditions required for germination of seeds. The following are 3 conditions for the seeds to germinate and develop into new plants. The first condition that required for germination of seeds is water. Water is needed to soften the testa and to activate the enzyme. The activated enzyme breaks down the stored starch in the cotyledon to glucose, which can be used in the respiration to supply energy for growth. The second condition is a suitable temperature. A suitable temperature is required to activate the enzyme to break down the stored starch. The suitable temperature for the germination is 10 degrees Celsius to 35 degrees Celsius. The third condition is the environment with enough air, the oxygen. Oxygen is required for cellular respiration. Oxygen enables seeds to respire to supply energy for growth. This is all the content of Topic 7, Plant Reproduction, and the explanation for Chapter 4, Reproduction, end here. Thank you for watching.